forget the drawing. But where one forms a head, the other cells form a foot, but they have the same DNA. It's all about controlling which of these genes that are on the cells in the cells, which are turned on and which are turned off. So the foot genes are going to have a certain sequence. The head genes are going to have a different sequence. And so the foot genes might have this one, and that one turned on. And maybe there'll be more of the head genes on one chromosome versus another. Maybe there'll be more head genes on certain chromosomes, but the point is that different genes are going to be turned on in different tissues. That's called differentiation. When you first were conceived, when your father's sperm met your mother's egg and they formed you were only one cell. You were a single cell. You started to divide 1 into 2, 2 into 4, etc., until you were a group of cells. During development, you turned into a group of cells, a little ball of cells called a blastocyst. And eventually, some of these cells started to change. They started, at first, when you were just this, and probably when you were just in this range of cells, we'd call these cells omnipotent. They could become anything. This cell could become any part of you, your brain, your eye, your foot, your liver, whatever. But soon these cells started to differentiate. Soon the head cells started to come together and the skin cells started to come together, and the cells were going to turn into blood and tissue, and blood vessels and such were going to come together. And so they started to differentiate. They started to become different. How would the cells become different? They would turn different genes on and different genes off. Does that make sense? So these cells lost their ability to become omnipotent. They no longer were omnipotent. They're not necessarily completely differentiated. So at some point, they became different tissues. Part became, brain. Part became an eye. Different parts of the eye, right? Because to this organ called an eye, there's different tissues. Some became muscles. Some became nerves. So once they became these different types, these different specific types, they're completely differentiated. So it's very hard for them to be anything else. That's why once you have a muscle in your hand, somebody cuts off your arm, once you're differentiated, once you're an adult, once you're a child, you've already made these decisions. These cells have made these decisions. They're turned off and they're turned on. Genes are turned off and genes are turned on. So that cell could only be a muscle cell. It can't become a bone cell. Your arm is not an arm cell, right? Your arm is made of skin cells and blood vessel cells and blood, right? And muscle and hair follicles and bone and and cartilage, right? So there's all kinds of different cells here. It's not just one cell type. So if I cut my arm off, these cells in my body would have to turn into bone and muscle and blood and skin, and they can't do that because they've already differentiated. They've already become different, and so they've lost their ability. They're no longer omnipotent. They're completely differentiated. There are some cells that keep their a level of omnipotence. We call them stem cells. Stem cells used to be just from embryos, embryos that were miscarried or aborted or whatever. And we're usually at this stage when they're one or just a group of cells here. But these stem cells are very important. You can get some level of potential. That's what potent means, right? Potential. Omni means all-encompassing, right? Potential, so they can become anything. Well, there's a type of stem cell you can get from the umbilical cord. That's one. There are other stem cells in your body, in your bone marrow and etc., but they've already lost some of their potential. But stem cells have been a very strong source of our research because they can allow us to become any part of your body. Now, we've come up with techniques that allow us to reset the DNA. We have methods now. We've done it with sheep and with other vertebrates, which when I was your age, scientists thought were going to be impossible. They thought anything more complicated than a frog wasn't going to be able to do this. But we found that if we do a series of techniques, we can undifferentiate. This undifferentiating process does come with issues. We haven't been able to perfect it yet. We can't clone you. But the hope is that we can take one of your skin cells or a cell from your leg, and if you need a heart, we can undifferentiate it at some point in the future through whatever process, and we can then grow you a new heart. We don't want to grow a clone. What would be the problem with just growing a clone? Where's some of the problems of growing a clone of you to harvest a heart or liver or whatever you need?
Wouldn't it be like deformities and stuff after?
Could be deformities and stuff because of the techniques, but let's assume we don't get rid of any of the problems in the techniques. We can grow a new clone. What's the problem? In fact, some people think that in China, some scientists in China said they've done it. So we can clone you. We can take and make a new copy of you. And then if you get sick as you get older, we can just change out your heart or your liver or whatever. What's the issue?
Would that person, the cloned heart, grow with you?
That's a good point. You have to wait till it grows up, right? So it's going to have to wait at least 18 years, right, for it to be fully mature. So that's no good because you're not going to have that much time probably. But let's say we can speed that up. So now we grew a whole clone. What's the issue? That thing has a brain. How are you going to take its heart for you? It's a living creature. It's a different creature. It's your twin, right? A clone is your twin. You're going to kill your twin so you can live? Oh, you're harsh. You would do well in Game of Thrones. But no, that is an ethical problem, right? So, but the solution is don't grow the whole clone. Just grow the heart. You need to learn these signals, though. It all comes back to these genes. We need to learn what those genes are that need to be turned on and which need to be turned off so that we can turn the right ones on and off so we can grow you an organ without having to grow a whole twin. Does that make sense? All right, so that would be the ultimate goal. By the way, this explains why a lizard can regrow a tail or a leg or an arm. Because lizards and geckos, for instance, and I don't know how many toes they have, but let's just say they look something like this. I know they don't, but whatever. So a gecko, you cut the gecko's arm off, it turns out that the gecko has some stem cells at the base of their arm and their tails. And if you cut them up, they have stem cells. They have that potentiality. They have potential, and they can regrow that arm. Other things are not as differentiated. There are some creatures that don't differentiate that much. Starfish, for instance. Starfish, you can take and chop a starfish in half, and it'll grow 2 starfish. Cut it up into 5 pieces, you'll grow 5 starfish. There's a limit, but that's because they don't have those many switches that they've turned on and off. Does that make sense? They're simpler, if you want to put it in a different way. And then you have a more simple organism called the planaria. Have you ever heard of a planaria? It looks something like that. It has little eye spots. Actually, kind of cute. But you can take those and you can and throw them in a blender, and you'll have a couple hundred planaria when they're done. They have each one of these has more potential, less differentiation, has some stem cells. This has more potential, even still, less differentiation, and this has the most potential, the least differentiated of these multicellular organisms as compared to humans. So the more complicated you are, the more specialized your tissues, the harder it is to go backwards, to go in the opposite direction, undifferentiate them, and get that potential. So my suggestion, you can talk to Miss Dalverin, if you ever talk to Miss Dalverin, she did this with her triplets. She had triplets, I don't know if you know that. What she did is she got her umbilical cord stem cells, and she had them saved. She had them frozen for her children in case that might help them in the future. So if you have babies, you can ask the doctors to save your umbilical cord stem cells, and they'll put them in liquid nitrogen. You could save them until your kids get old, in case they might be able to use some of those for some of these techniques that I've just been talking to you about. Does that make sense to all of you?
Mm-hmm.
All right. And then that would get rid of the idea of, because a lot of people have a problem with stem cells because some of the stem cells might have come from abortion clinics, etc. That would solve your problem, wouldn't it? If you saved your own umbilical cord stem cells. All right. So now let's talk a little bit about what we've been talking about all this time, which is proteins. How do we go from a gene to a protein? How much time do we have left?
It's
8:32. So we got some time. So we're talking about going from a gene. Where is this gene? Where is this? This thing is called, by the way, let's talk about what this thing is called. This is called, anybody know what you would call this thing? DNA molecule. Good. We say it's double-stranded, but a chemist would argue that these are actually 2 molecules, hydrogen bonded together. I'm glad you can see that, because a chemist would argue that these are 2, and there's a really good point to that. They are, because these are just hydrogen bonded to one another, and there's 2. There's a 3' end and a 5' end. And then they're just hydrogen bonded to one another. 3' and a 5'. And this is 5' and this is 3'. So they're complementary, they're hydrogen bonded, they're double helix, they form, but a DNA, we call it a DNA molecule just to make it simple and easy. That's why they call it a DNA molecule. So this is very long. If you looked at when DNA in your cells are all wrapped up, let me redraw this. If I drew your cell, if I looked through a microscope at any one of your cells, and we had a really good microscope and we knew what we were doing, you would see a lot of these structures. You'd probably see a few dots that would get. Then you'd see this. If you zoomed in to your nucleus, that's where you'd find your DNA. You'd find your DNA, and I'm going to go ahead and draw the DNA as blue. You would see your DNA would look like this most of the time. it would look like a bunch of spaghetti. We call that chromatin. Chromatin. That's when the DNA is all unwrapped. It's all unwrapped, it's being used. It's when DNA is being functional. It's doing its job when it's unwrapped. So when it's most of the cell's life, the chromatin is all unwrapped. That ball of spaghetti is actually 23 pairs of DNA. One set of 23 from mom and 23 from dad. So for a total of what? 46. 46 chromosomes and 46 copies in humans. This is in humans, obviously. I hope you know it's in humans. In humans. Corn has hundreds. Corn has hundreds of these copies. There are some organisms that only have 3. So the number of chromosomes depends on the creature, on the species. So 46 copies. When not all of the DNA is unwrapped. Some of it's unwrapped, then some of it's wrapped up around histones. I'm going to go ahead and draw the histones in red. And then some more unwrapped, and there's some more around histones. What do you think is going on here? Why is some of it unwrapped and some of it not unwrapped? Yeah.
Some of it is being used like at the moment.
That's right. Excellent. So some of this DNA is being used. So this is used. That's excellent. That's really that shows you've been paying attention. That's really a good point. This is being used, right? All the stuff that's unwrapped is being used. So what about the stuff that's wrapped up? What about the stuff that's wrapped up?
It's not being used.
It's not being used. It's being stored. So we call it off, right? Not used. And so these are genes that are off. So these are the genes. This is how one way we can turn genes on and off is to keep them wrapped up around histones. We keep them put away. We're not using them, we're storing them. The ones that are being used are unwrapped. And you unwrap different sections of the DNA depending on which cells you're talking about in your body. But the DNA molecule, the sequence from one end to the other is the same. It's just your eyes, you wrap up certain ones, and in your foot, you wrap up another section of it. Does that make sense? That's just one way. There's several ways we turn genes on and off, but that's one big way. Are we good with that? Mm-hmm. All right, so there's 23 pairs, and again, you get 23 from mom and 23 from dad. And we'll talk more about that when we get into genetics next week. Next week, we're already in genetics, so. All right. This week, what you got to understand is in the nucleus, all in the nucleus, this is all happening inside the nucleus. DNA, and please know this by heart, is only in the nucleus. In eukaryotic organisms, I hope that's obvious. I hope to me, they seem obvious. But let me be very clear. In organisms with a nucleus, DNA is only in the nucleus. Did I make that statement clearly enough? Mm-hmm. Prokaryotes have DNA. This is a mistake that students make all the time. They think DNA is only in the nucleus. Prokaryotes don't have nucleus, so what do you think? They don't have DNA. Well, that's not true. They do have DNA. It's just not in the nucleus, okay? So don't get that confusion in your mind. Those organisms that have a nucleus, the DNA is in the nucleus. When it's being used, it's unwrapped. It's formed these things called chromatin. And again, the DNA is only found in the nucleus. Now, I should tell you about 2 other kinds of DNA. When we're ready to move and separate the cells, what do you do when you're ready to move stuff? You're working in a law office, and the law office has thousands of files and folders and cabinets all around. You have to organize them, and you're going to move offices. You're a lawyer, and you're going to move to another building. What do you got to do?
Pack them up.
You have to pack them up, right? Otherwise, you're going to lose them. You're going to lose them. Things are going to get confused, right? That's no good. So we have to take all this ball of spaghetti, and we have to make copies of them, because we're going to split them into two, right? So we have to make copies of them. And then we have to hold them together, make sure we keep them together, make sure we keep them straight so we can move them. So we need to pack them up. Well, when we pack them up, when we make copies and pack them up, this is what they look like. They look like this. They're packed up. They're made copies. These are copies of one. These are copies. They're stuck together. Keep the copies stuck together. They're like paper clips. Keep the copies together. And they're all packed up. They're no longer unraveled. They're 100% wrapped up. So they're wrapped up around histones in this tight little, sort of like braiding your hair, right? They're tight braids around, and they actually get real tiny, so tiny that you get 46 of these times 2, because there's 2 of them. They're copied, so there's 46 of these. We call these chromosomes. It's the same DNA. The DNA didn't change. They're just packed up. When you hear the word chromosome, you're just talking about the fact that you're wrapping it up. And you only see these wrapped up chromosomes after the copies. So you have 2 copies. So technically, you don't have 46 anymore. You have 92. And I shouldn't have said, so you start off with 2 copies of each chromosome. See, this is technically, each one of these is a chromosome, but when they're together, we also call them a chromosome. So together they're a chromosome. Once they separate, we'll also call them a chromosome. So don't get confused about that. You'll see that when we talk about mitosis and meiosis. But these 2 copies here, these 2 copies are called sister chromatids. We'll talk more about these later, but it's, I think, a good time to discuss these different terms. Yeah.
Isn't that one meiosis and the other one mitosis has like 4? Isn't it something like that?
Yeah, it's something like that. It's mitosis happens. We'll talk about it. I guess it's really kind of cool. I like mitosis. You should have learned a little bit about it in middle school, probably, if you had a chance, if you had a teacher that liked it. Mitosis and meiosis are very important. Mitosis helps you repair yourself when you get cut. Mitosis allows you to repair your cells. They replace cells that you hurt or damaged. Meiosis makes when you make a baby, you're doing meiosis to make your eggs. You have 10,000 eggs. Those eggs all were formed from meiosis. You take half the DNA. You don't have 46 copies in your eggs. You only have 23 copies. Only half the DNA. That makes sense if you think about it. If you're going to get together with your husband, you're going to make a baby, you're going to need... That's right. Excellent. 23 of his and 23 of yours, and you make one whole person, right? All right. But that's only for humans, because different ones and different creatures. All right. So unwrapped, they looks like that, and wrapped up, they look like that, and that's how you turn them off. So now the question is, what's happening when you're turning them on? You're taking this sequence. I'm just going to write a little, just a quick example. Let's say it's A, T, G, C, A, T. That's the sequence. It's a lot longer. It could be hundreds, maybe even 1,000 nucleotides long, because these are very long. Understand, they're only a single molecule, but it could be as long as 3 meters. So it's really long. And then, of course, you wrap them up into this tiny little, all of it's all in one nucleus. So you can imagine how tiny these things are. So the first thing you do is what? What do you do? Oh, I should tell you. When you're talking about it, not all of the DNA is a gene. Only a certain section, only this part right here is going to be a gene. If this is the gene right here, if this is the sequence that's going to be turned, and by the way, when we're expressing a gene, when we're turning a gene on, when we call it a gene, that means this turns into an RNA. We don't call it, they're not genes unless they make RNA. Is that clear? Mm-hmm. All right, so the rest of the DNA is used to regulate. So you have this area before the gene. This area before the gene is called the promoter region. What do you think the promoter does?
We learned
It's
about
all-
it
I heard
Yeah, you probably saw it yesterday a little bit. What do you think this little section of pink does here, this promoter area?
It helps run.
It helps, it promotes, it increases. Without the promoter, it's not going to happen. So I like to think of the promoter area as kind of the landing strip. To make anything happen, what is it that you need to have almost anything happen in a cell? Protein, a specific kind of protein called what? Well, they could be ribosomes, which are a mixture of proteins and RNA. But specifically, it's an idea with the enzyme. That's right. There's this enzyme complex. It's a complex. There's more than one thing. It's called RNA, because this is what it's going to make. It's going to make RNA. Polymer, because it's going to make a polymer. A polymer of what? RNA. ACE. All right. ACE means it's an enzyme. So you got this polymerase. It's called RNA polymerase. It doesn't really look like this, but it looks something like it's just this blob of proteins, different polypeptides. They need to come together and need to land here on the promoter region. So I just think of this as a landing strip for the enzyme. So the RNA polymerase comes in, lands on the promoter region, and then starts to read the DNA. The DNA has to open, so you get this thing that looks like this. There's two strands, so they have to open. And then there's a coding side, the side that's going to give you what that's going to be read. And then there's a non-coding side, the side that's not going to be read. So this is going to be read, and this is where you're going to see A, T, G, C, A, T. Now, if you looked at the opposite side, this would not be A, T, G, C, A, T, right? What would this side be? T, A, C, G, T, A, right? So this is the coding side. So that when the RNA polymerase lands and starts reading it, it's going to start making RNA. So this is going to start making RNA. So the first thing that happens is you get these things called transcription factors. That's another way we can control what turns on and what turns off. These little proteins in different RNA sections can land, and they're called factors. They're called transcription factors. This whole thing, taking DNA to RNA, is called transcription. It's called transcription. And these transcription factors, what they do is they help turn on those genes. Because you don't want these genes just turned on all the time. That would be a waste and also would be confusing for your body. You can't have every chemical reaction happening all the time. The whole point of life is organization. The whole point is you want to feel love when you want to feel love. You want to be angry when you're angry. You don't want to feel angry and love, hate, fear, bravery, right? You need to have one thing at a time. If you're feeling everything at once, you're just convulsing on the ground. You're not any use to anyone. Does that make sense? So you can't just go and stop. You can't just run and sit. That can't happen. So you have to have one thing happening at a time. So you need to control which ones of these genes get turned on and off. By the way, that's a really bad example because all those things were not directly controlled by a gene. But I'm just saying when you're trying to do a thing, you only want to do one thing at a time. You don't want to do everything at once. Does that make sense? So I use those examples. All right. And then at the end, you have what's called the termination sequence, right? So you have promoter, you have the gene, and then you have term. So the termination sequence, that turns it off. So So here's where the RNA polymerase lands, and this is where it gets kicked off. Lands, kicks off. Lands, kicks off. This middle part is what's being read. Then you read it, and you turn it into an RNA. Did I say mRNA?
No, you said
No,
RNA.
I said RNA. There's a reason I said that, so pay attention. So when I make this, it's going to be RNA. How would you transcribe this into RNA? It would be, would I make it
Opposite of
opposite A, which would be what?
T.
No. No T in RNA. No. Can't get matched with T. What does it get matched with? No. C only gets, with U. In RNA, you only form U. T takes the place of, or U takes the place of T in RNA. Right? I know it's a long time ago that we talked about it, but it's important that you know that. So you got U, then T gets read to what? Opposite of T is what? A.
Just U takes T's place.
U just takes the T's place. It doesn't take the A's place. So there is A in RNA, but there's no U. There's no T, right? So UA, then what? The G gets turned to C, and the C gets turned to
G.
And the A gets turned to
U.
And then the T gets an A. All right, and it goes on. It's a long piece. This is not mRNA yet. This is RNA. Then there's further control. Like there are other changes that have to happen to this RNA, and some of the things that have to happen is the introns have to be taken out. So there's sections of this that you're going to cut out. They're called introns, which never made sense to me because I always thought introns you should leave them in.
In because it says in.
In, right? But it's opposite what I think, but that's how I remember it. It's opposite what you think, Mendoza. That's how I remember it. Exons everything else is called an exon, and those exons, they stay. They get spliced together. There's a whole process we're not going to get into. There's a whole process. It takes the two sections that are not introns and it puts them together and makes one whole piece. And that piece has the U, the A, the C, the G, the U, and the A, and all the rest without the introns, minus the introns. It's still not quite RNA because the last thing we have to do is we have to add a poly A tail. We have to add a bunch of A's at the end. Once you add the poly A tail, you've taken out the introns, you add the poly A tail. Now this is an mRNA.
So mRNA is when it has the poly A tails?
It has a poly A tail and the introns have been taken out. Now I got to tell you, not all genes go to mRNA. Some genes get turned into RNA. Some genes, so let's take a quick look at what genes do. So yes, a lot of genes do go to mRNA. You can go to mRNA. We just went through the process. By the way, that's it. That's the end of transcription. That's all there is to it. Not that big a deal. Translation's a little more to it, and that's just taking a ribosome. We'll do translation tomorrow, and it's kind of fun. Most of the kids like doing it. It's like a puzzle. You have to read a chart, and just as easy as it is to go A to U and G to C, it's going to be very easy to read mRNA as well. So genes can go to RNA, mRNA, or they could just go to RNA alone. Things like tRNA, they can form ribosomes, and other structures that don't actually go to mRNA and make proteins. Remember, from mRNA you go to proteins. That's just a major path of genes. Not all genes go to mRNA and then to proteins.
So
All right.
what happens if it's not in that specific order?
Say that again.
What happens if it's not in the specific order of U, A, G, C, G? What would happen? Would it form something different?
Yes, absolutely. If you mess this up, you have a mutation. If it's here in the DNA, if you messed up the sequence here in the DNA, you have a mutation.
So, on RNA, the introns are still in there.
In regular RNA, when it's first transcribed, you still have the introns. Then something called a spliceosome comes in, cuts them out, and splices them together. Then adds a poly A tail. Now you got an mRNA. Now you're ready to make a protein. Hey, what's up?
Are they here to me Finley and
Taylor
Taylor Hudson?
Hudson and who?
Tyler.
I know. I'm sorry.
Yeah.
She said Taylor. I didn't. What is the other one?
To me Finley?
Finley. What do you need?
They just have to fill out a one-question survey for stats.
You have to fill out a one-question survey for stats. Are you here? Go to her.
It's time for us to go to the
All right, my friends, have a good one. You, Big Mouth, come here. What did you find during your research? I'm interested.
A lot.
Did you find anything that makes you think at all?
Some
Is
of it.
Some of it, yeah. Yeah, I found when I researched anti-vax positions, I also found it very interesting. There's some points that you need to consider when you're making those decisions. You know what I'm saying? But I want you to make I just play with you in class, but I just want you to think about it when you're making decisions. Don't listen to people. People are fools. When you talk, you got to talk about facts and then make decisions for yourself. All right, have a good one. What happened to Emily? Where's Emily? What's going on? They left.
All of them did. She was here today.
Well, there's a whole drama going on. I don't know what's going on, but I have to find out. I'm absent a few days, and everything gets turned upside down.
That's what always happens.
That's why I hate being absent. I'm doing all right. How you doing? Yeah, what's up?
So I figured I found out.
Don't be talking to me without a mask. Get away from me.
Bro.
Be 6 feet away from me.
Video on YouTube. Yeah, he gonna post it.
I'll try to post it today, yes.
You okay?
It's all right. It's hard, but you know how it is. I mean, I don't know if you know, but it's hard.
What? Why left my grandpa?
I hope you don't know. I hope you don't know for the many, many years to come.
I'm sorry.
It's all right, but listen, you're great. Make up everything you can. Just get it done. All right. If you need help, I'll be here during
I gotta
lunch.
be. I just need to.
Which lunch though?
The second lunch,
It's just
also
the
after
debate
school.
Stuff. That's the only thing I need to do. I did everything.
Debate stuff. We'll get this stuff done. Turn it in.
Did
I'll grade it. I don't care anymore. I'll grade it.
Did you see what I sent you?
No.
I had sent you a message because about my lab report. I turned it in with the first revision.
Why don't you see me during second lunch or after school? Quick though, because I need to go and get some stuff ready for the funeral.
I found out some more facts about the debate.
Oh, excellent. Well, I hope you're ready, baby.
She works for a hospital.
Excellent, but don't be late to class. See me after school, okay, baby?